Everything you have been told about the giant isopod is probably wrong, not slightly wrong. Not a matter of dusty textbooks that just need a little updating. Wrong in a way that is fundamental. Wrong in a way that, once you understand it, makes you look at the deep ocean differently, and maybe makes you look at life on this planet differently too. For over a century the story was simple. The giant isopod was a scavenger, a bottom feeder, a glorified roly-poly that got big because the ocean let it. It sat in the dark on the seafloor, thousands of feet below the sunlight, and waited for dead things to fall from above. Whale carcasses, dead fish, the slow rain of organic debris that marine biologists call marine snow. That was the narrative. A patient, passive creature doing a thankless job in the coldest, darkest corner of the planet. The ocean's janitor, the deep sea's clean-up crew, and it made sense. The giant isopod looked like a creature designed for that role. Pale lilac armor plating, 14 legs, 4 sets of jaws, compound eyes the size of marbles with nearly 4,000 individual facets, each one backed by a reflective layer called the tapetum lucidum, the same structure that makes a cat's eyes glow in the dark. It looked ancient because it was ancient. It looked passive because, well, we assumed it was. But in the last few years that story has started to unravel, not slowly, not quietly. It has come apart in ways that have forced marine biologists to reconsider almost everything they thought they knew about this animal. In 2022, a team of geneticists sequenced the giant isopod's genome for the first time. What they found was staggering. The genome was nearly twice the size of a human being's, packed with genetic machinery, specifically designed for extreme growth, extreme fat storage, and extreme digestive efficiency. It wasn't a passive scavenger that happened to be big. It was an organism whose entire genetic architecture had been engineered over hundreds of millions of years for precisely the life it leads. That same year, a different team of researchers discovered that a specimen sitting in a Japanese aquarium, classified for years as Baphinomus giganteus, the most well-known giant isopod species, was actually something else entirely. A new species, hiding in plain sight, in an aquarium, for years. Nobody had noticed. And then there was the footage. Deep-sea camera footage of a giant isopod latching onto the face of a living dogfish shark and eating it. Not scavenging a dead shark. Not nibbling at something already gone. Actively attacking a living, struggling animal larger than itself and consuming its face. We were wrong about the giant isopod. Not about one thing. About almost everything. And if we got this animal so wrong, despite over 140 years of study, despite examining thousands of specimens, despite it being one of the most recognizable creatures in the deep ocean, then it is worth asking what else we might be getting wrong about the world beneath the waves. To understand how we built such a misleading picture of this animal, we need to go back to the very beginning. To 1879, to a French zoologist, a specimen jar, and a creature pulled from the darkness of the Gulf of Mexico that shocked the scientific world. In 1879, a French zoologist named Alphonse Milne Edwards received a specimen that had been collected by Alexander Agassiz from fishermen operating near the Dry Tortugas, a small group of islands at the western end of the Florida Keys. Agassiz had been conducting deep-sea surveys in the Gulf of Mexico, lowering nets and dredges into waters that, at the time, most educated people assumed were essentially empty. The prevailing scientific consensus of the mid-19th century held that the deep ocean was azoic, lifeless, a vast, cold, pressurized void where nothing could survive. This idea had been challenged in recent years by the work of Sir Charles Wyville Thomson and the Challenger expedition, which had begun to pull strange organisms from great depths, but it had not yet been fully overturned. The deep sea was still largely a mystery. And into that mystery, Alphonse Milne Edwards now held something extraordinary. The specimen was a juvenile male. Even as a juvenile, it was enormous by isopod standards. To put this in perspective, isopods are a group of crustaceans that most people encounter in their gardens. woodlice, pillbugs, roly-polies, those small grey armoured creatures that live under logs and in damp corners. The average woodlouse is about 1.5 centimetres long. You could fit one on your thumbnail. What Milne Edwards was looking at was a deep-sea relative of those creatures that could grow to over 76 centimetres in length and weigh nearly 2 kilograms. That is roughly the size and weight of a small dog, except it was an isopod, a pillbug, from the bottom of the ocean. Milne Edwards described the specimen and established the genus Baphonomus, placing the new species as Baphonomus giganteus. The name was appropriate. The discovery was a sensation. Newspapers picked it up. The public was fascinated and a little horrified by the idea that a creature resembling a giant armoured woodlouse was living in the permanent darkness of the deep sea, thousands of feet below the ships that crossed the Gulf of Mexico. But from the very beginning, the way we studied this animal created a problem, because we couldn't visit the deep ocean with anything approaching regularity in the 19th century, and because submersible technology was still decades away. Everything we learned about the giant isopod came from dead specimens, animals pulled from trawl nets, animals collected from baited traps, animals that arrived on the surface already dead, their bodies often damaged by the pressure changes involved in hauling them, from depths of caisson 600 to 2,000 meters. From these dead specimens, scientists built a picture. They examined the anatomy. They noted the compound eyes with their thousands of facets and the reflective tapetum, which suggested the animal could see in near total darkness. They counted 7 pairs of walking legs, 4 sets of jaws and 2 pairs of antennae that could stretch half the length of the body. They observed that the animal could curl into a ball, just like its garden-dwelling cousins, tucking its legs and soft underside beneath a dome of armoured plates. They opened the stomachs and found the remains of fish, cephalopods, shrimp, and squat lobsters. They found evidence of marine snow, the constant gentle drift of dead organic material from the sunlit surface waters to the dark bottom. And from all of this they drew what seemed like a reasonable conclusion. The giant isopod was a scavenger, a deep-sea garbage disposal. It sat on the bottom, waited for food to arrive, ate what it could, and survived by being patient and efficient. And that was the story for over 100 years. 143 years, to be exact. The giant isopod appeared in textbooks, documentaries, and museum displays as a classic example of deep-sea scavenging. It became one of the most recognizable animals in deep-sea biology, precisely because it fit so neatly into the narrative we had constructed. It was big, it was slow, it was ancient, and it ate dead things. Simple. But there was a problem with this story. Actually, there were several problems, and they all trace back to the same fundamental issue. We were studying dead animals and making assumptions about living ones, when you can only examine specimens that have been pulled from their environment, crushed, chilled, and preserved. You are working with a snapshot. A single frozen frame from what is, in reality, a continuous, complex, active life. You can learn a great deal from anatomy. You can learn what an animal is built to do, but you cannot learn what it actually does, not with any certainty. You cannot observe its behaviour. You cannot watch it interact with its environment. You cannot see it hunt, or fight, or hide, or reproduce. You are left with inference. And inference, even careful inference, is a guess. For the giant isopod, the guess is accumulated into a consensus, and the consensus hardened into fact. Scavenger. Passive. Simple. A big roly-poly that waits for dead things. And for over a century, nobody had a strong enough reason to question it. The first real crack in that consensus came not from the isopod itself, but from a question that nobody could satisfactorily answer. Why is it so big? This is not a trivial question. Deep-sea gigantism, the tendency for deep ocean species to grow far larger than their shallow water relatives, is one of the most striking and persistent patterns in marine biology. The giant squid, the Japanese spider crab with a leg span that can exceed 3.5 meters, the giant oarfish which can grow longer than a school bus, the giant tube worm, which lives near hydrothermal vents and can reach over 2 meters in length. All of these animals are dramatically larger than their nearest relatives in shallower water. For decades, marine biologists offered several theories to explain this. The most common was cold temperature. Colder water increases cell size and, in organisms like crustaceans that continue growing throughout their lives, extends their lifespan, allowing more time to accumulate mass. This follows a principle called Bergmann's rule, the same observation that explains why polar bears are larger than bears in warmer climates. And there is evidence to support this. In Arctic and Antarctic waters, where the temperature difference between the surface and the deep is relatively small, the size gradient with depth is also reduced. Cold water does seem to enable larger bodies. Another theory pointed to food scarcity. The deep ocean is oligotrophic, meaning it has very low nutrient availability. Food is rare, meals are unpredictable. In such an environment, an animal that is larger has a lower metabolic rate per unit of body mass, following a principle called Kleiber's law. A bigger body burns energy more efficiently than a smaller one. This means a large animal can survive longer between meals. Giant isopods are a vivid illustration of this. They gorge themselves when food is available, distending their bodies so massively that they can barely walk, and they can survive extraordinary periods without eating. One specimen held at the Toba Aquarium in Japan refused food for over 5 years and 140 days before it finally died. 5 years without eating. That is not a quirk. That is an adaptation refined by deep evolutionary pressure. A third theory invokes reduced predation. The deep ocean is dark and sparsely populated compared to shallower waters. There are fewer predators. This means less evolutionary pressure to stay small, fast, and inconspicuous. A large body becomes viable when the things that would eat you are rare, and giant isopods do have few natural predators. Their exoskeleton is thick and reinforced with calcium, and there is proportionally little meat inside compared to, say, a crab. Most predators simply do not find them worth the effort. A fourth theory, less commonly discussed but worth mentioning, involves dissolved oxygen. Certain zones of the deep ocean contain higher concentrations of dissolved oxygen. Since the size of land-based invertebrates is fundamentally limited by their ability to absorb oxygen through passive diffusion, the argument goes that aquatic invertebrates in oxygen-rich deep water may face fewer constraints on how large they can grow. All of these theories have merit. All of them are probably contributing factors, but none of them, individually or combined, actually answer the question. They explain why being big in the deep ocean might be advantageous. They do not explain how the giant isopod's body actually achieves it. They describe the benefits of the building, not the engineering that made it stand. It is like saying that a skyscraper is tall because tall buildings are useful in cities with expensive real estate. True, perhaps, but it tells you nothing about the steel, the foundation, or the architecture. For over a century, the mechanism of deep-sea gigantism in isopods remained opaque. We knew the why at a surface level. We had no idea about the how. And then, in May of 2022, a team of researchers led by Jumbo Yuan at the Chinese Academy of Sciences cracked open the blueprint. And what they found inside was stranger and more sophisticated than anyone had anticipated. The specimen was a Bathynomus jamesi, collected from 898 meters deep near Hainan Island in the South China Sea. Yuan's team sequenced its entire genome using a combination of long-read and short-read technologies, assembling the most complete genetic map of any giant isopod ever produced. And the very first thing they noticed was the size of it. The genome was enormous, 5.89 gigabases. For context, the human genome is approximately 3.1 gigabases. This isopod, this deep-sea crustacean that we had been calling a simple scavenger for 140 years, carries nearly twice as much genetic information as a human being. It was, at the time of publication, the largest sequenced crustacean genome on record. but the sheer size of the genome was only the beginning of the surprise. The real story was in the composition. 84% of the genome consisted of transposable elements, sequences of DNA that can copy themselves and move to new positions within the genetic code. Scientists sometimes call these jumping genes. In most organisms, transposable elements make up a much smaller fraction of the genome. In the giant isopod, they dominate it. This matters because transposable elements are linked to high rates of mutation and what geneticists call genomic plasticity. They make the genome flexible, adaptable, capable of responding to environmental pressures in ways that a more rigid genome cannot. Yuen's team proposed that this extraordinary proliferation of jumping genes may be one of the reasons the giant isopod has survived and adapted so successfully over such immense stretches of geological time. But the most striking findings came when the researchers looked at what specific genes were doing. They found expanded gene families related to thyroid hormone signaling and insulin hormone signaling, two pathways that are directly involved in growth regulation and nutrient absorption. In plain language, the isopod's genes are specifically tuned to make it grow larger and to extract more nutritional value from every meal. These were not incidental findings. The expansion was statistically significant compared to other crustaceans, including the giant isopod's much smaller terrestrial cousin, the common woodlouse. They also found genetic modifications that suppress the breakdown of fat. Giant isopods store enormous reserves of lipids in a structure called the fat body, which is distributed throughout their body cavities. The primary form of stored fat is triacylglycerols, which make up 88% of the lipids in these tissues. The genome study revealed that this fat storage is not merely passive accumulation. The isopod's genes actively slow the rate at which stored fat is metabolized. They hold onto their reserves the way a miser holds onto gold. Every calorie is precious, and the genome is designed to make each one last as long as physically possible. And then there was the stomach. Anatomical examination of the specimen showed that when fully expanded, the stomach occupies 2/3 of the entire body cavity. 2/3. Imagine an animal whose stomach fills most of its body. The genome revealed expanded gene families for glycolysis and vesicular transport that were specifically expressed in the digestive organs, meaning the gut is not just large but genetically optimized for maximum extraction when food is available. So the full picture that emerged from the genome was this. The giant isopod is not an animal that happens to be big because the deep ocean allows it. It is an animal whose entire genetic architecture has been deliberately refined over hundreds of millions of years for a specific mode of existence. Expanded growth hormones to build a larger body. Suppressed fat metabolism to preserve energy reserves. A stomach that fills most of the body cavity. Backed by genes that maximize nutrient extraction from every meal. It is a biological machine designed from the DNA up for feast or famine, survival at extreme scale. The phylogenetic analysis placed the divergence of deep-sea and terrestrial isopods at approximately 245 million years ago. Fossil records show that Bathynomus itself dates back at least 160 million years. These genetic adaptations are not recent. They have been refined across geological time, shaped by hundreds of millions of years of unrelenting deep-sea pressure. This was the answer to the question that nobody could answer. Not cold water, not reduced predation, not dissolved oxygen alone. The how of deep-sea gigantism in isopods is written into their DNA, and it is far more complex, far more engineered and far more purposeful than anyone had guessed. But the genome was not the only surprise to emerge in 2022. While geneticists were rewriting the biology, the isopod's behavior was also demanding a complete rewrite. And that rewrite started with a piece of footage that, once you see it, you cannot unsee. For over a century, every scientific description of the giant isopod emphasized the same word, scavenger. The textbooks said it, the museum placards said it, the documentary said it, And yes, giant isopods are scavengers. That much is not in dispute. They eat dead things. They are very good at eating dead things. When a whale carcass settles to the ocean floor, an event marine biologists call a whalefall, giant isopods are among the first creatures to arrive. They have been observed gorging on dead fish, dead squid, and the bodies of other crustaceans. One of the most comprehensive studies ever conducted on their diet examined the stomach contents of 1,651 specimens of Biphanemus giganteus, collected off the Yucatan Peninsula in Mexico. The researchers found that fish were the most commonly consumed food, followed by cephalopods and decapod crustaceans, particularly shrimp and squat lobsters. Classic scavenging, classic bottom feeding, the textbook picture confirmed. But the same study found something else, something that did not fit so neatly into the scavenger narrative. Mixed in with the remains of dead fish and dead squid, the researchers found evidence of predation on living organisms: sea cucumbers, sponges, tunicates, echinoderms. These are not dead things drifting down from above. These are living animals on the seafloor, slow-moving, yes, defenseless mostly, but alive. Now, you could argue that eating a sea cucumber is not exactly the same thing as being a predator. Sea cucumbers are not fast. They do not fight back. They are, to put it charitably, among the least difficult prey items on the planet. But the point is not that the giant isopod is hunting gazelle. The point is that the label scavenger was incomplete. It was always incomplete. And the evidence that it was incomplete was sitting in those stomach contents in a study published in 1991, for over 30 years before anyone really grappled with the implications. And then came the footage. During a 2015 episode of Discovery Channel's Shark Week, titled Alien Sharks, Close Encounters, cameras placed near a deep water baited trap captured something that changed the conversation entirely. A giant isopod latched onto the face of a living dogfish shark and began to eat it. Let that image settle for a moment. A crustacean, approximately the size of a football, attached to the face of a living shark, a shark that was larger than the isopod, a shark that was still alive and struggling, and the isopod was eating its face, not scavenging a dead shark, not nibbling at an already decomposing carcass, actively aggressively feeding on a living predator. The footage was startling, because it contradicted everything the textbook said. Giant isopods were passive. They were patient. They waited for food to come to them. They did not attack living animals, certainly not living sharks. And yet there it was. On camera. Undeniable. This single piece of footage does not mean that giant isopods are apex predators. They are not. They do not hunt sharks as a regular part of their diet. The dogfish in the footage was likely weakened, possibly already injured by the trap. The isopod took advantage of an opportunity, and in the deep ocean, opportunity is everything. But that is exactly the point. The distinction between scavenger and predator, which seems so clean and obvious in a textbook, dissolves in the deep sea. When you live in an environment where food is scarce and unpredictable, where meals might arrive once every few months, or even once every few years, you do not have the luxury of dietary specialization. You eat what you can, when you can, however you can. If that means stripping the flesh from a dead whale, you do that. If that means consuming a living sea cucumber, you do that. And if that means latching onto the face of a living shark and eating it while it struggles, you do that too. The giant isopod is not a scavenger. It is not a predator. It is an opportunistic omnivore operating in one of the most resource-poor environments on the planet. It has 4 sets of jaws designed for ripping through tough biological material. It has a stomach that can expand to fill 2/3 of its body. And it has a metabolism so efficient that it can survive for over 5 years without a single meal. This is not a passive animal. This is an animal that has been honed by hundreds of millions of years of deep-sea evolution into one of the most efficient feeding machines in the ocean. The fact that it is usually still and usually patient does not make it passive. It makes it calculated. Think of it this way. A lion resting under a tree in the African savannah spends most of its time doing absolutely nothing, sleeping, lounging, conserving energy. If you only ever observed the lion at rest, you might conclude that it was a passive, lazy animal that spent its life lying in the shade. But the lion rests because hunting is expensive. It conserves energy so that when the moment comes, when the opportunity presents itself, it can explode into action with devastating efficiency. The giant isopod operates on the same principle, just stretched across a timeline that would make a lion weep. Instead of hours between hunts, the isopod waits months, years, and when food appears it does not hesitate. The behavioural rewrite did not stop at the predator question. It extended to something even more unsettling. The realization that we had been looking at this animal for over 140 years and had not even correctly identified how many species we were looking at. In 2017, a giant isopod was collected from a baited trap off the coast of Mexico's Yucatan Peninsula. The trap had been set at a depth of between 600 and 800 meters in the warm, dark waters of the Gulf of Mexico, where Bathinomus giganteus had been first discovered in 1879. The specimen was purchased by the Enoshima Aquarium in Fujisawa, Japan. It was classified as Bathinomus giganteus. Just another giant isopod. Nothing unusual. It was placed in a tank fed, maintained, and displayed alongside other deep-sea specimens. Nobody looked twice. Then a researcher named Huang Mingqi from Taiwan's National University of Tainan obtained the specimen for a project on isopod genetics. Huang was sequencing the DNA of various Bathinomus specimens as part of a comparative study. It was routine work. He took tissue samples, extracted DNA, and ran the sequences through standard molecular analysis, comparing the genetic markers of the Enoshima specimen against the known genetic profile of Bathinomus giganteus. The results were not routine. The genetic sequence showed at least 35 differences between this specimen and Bathinomus giganteus. 35. That is not a minor variation. That is not a regional population difference. That is a different species. Huang brought in 2 additional experts, Tadashi Kawai, the president of the International Association of Asticology, and Neil L. Bruce, an honorary researcher of invertebrates at the Queensland Museum of Australia. Together, they conducted a thorough morphological examination of the specimen. They measured its body proportions. They counted its spines. They examined its antennae, its legs, its shell. What they found confirmed the genetic evidence. The specimen was distinct from Bathinomus giganteus in multiple ways. It had more slender body proportions. It was shorter in total length, approximately 26 centimeters. Its antennae were longer. Its shell had a distinctive creamy yellow colour, different from the pale lilac of B. giganteus, and its thorax was shaped like an inverted triangle rather than the broader oval typical of the known species. In 2022, the team published their findings in the Journal of Natural History. The new species was named Bathinomus yucatanensis, after the Yucatan Peninsula where it had been collected. It became the 3rd species of giant isopod identified in the Gulf of Mexico, joining B. giganteus and B. maxiorum, which had been described in 2016. But the truly remarkable part of this discovery was not the new species itself. It was what the researchers wrote in their discussion section. They noted that over 1,000 specimens of Bathinomus giganteus had been studied over the course of 143 years. 1,000 specimens, examined by dozens of researchers across multiple institutions in multiple countries over more than a century. And nobody had noticed a second species living in the same waters with the same number of tail spines. The researchers were blunt about this. They wrote that there is a long history of misidentification of species in the genus. They pointed out the superficial examination, using only the number of pleotelson spines on the tail, could easily result in specimens of B. yucatanensis being misidentified as B. giganteus. The two species share the same spine count. The differences are in the body proportions, the antenna length, the shell colour, and the shape of the thorax. Differences that you would only notice if you were specifically looking for them. And for over a century, nobody was. This raises an uncomfortable question. If a distinct species of giant isopod could hide in plain sight for over 100 years, sitting in an aquarium in Japan, being handled and examined by trained biologists, how many other species might we be missing? The researchers themselves suggested there may be additional undiscovered Baphonemus species in the Gulf of Mexico and the broader Atlantic. They noted that many regions of the deep ocean are poorly sampled. The greatest known species richness is found off eastern Australia, where 5 species have been identified. But as the researchers cautioned, it is entirely possible that other regions, less thoroughly surveyed, harbour equal or greater diversity. Currently, the genus Baphonemus includes roughly 20 living species distributed across the Atlantic, Pacific, and Indian Oceans. No known species occurs in both the Atlantic and the Indo-Pacific. Previous records of B. giganteus from the Indo-Pacific are now considered misidentifications. The distributions of giant isopods are, by the researchers' own admission, imperfectly known. We are still discovering what is out there, and we are still correcting mistakes we made a century ago. There is something deeply humbling about this. We tend to think of taxonomy, the science of classifying organisms, as a sole problem for large, well-known animals. We accept that there might be undiscovered insects in the Amazon or unnamed microbes in the soil, but giant isopods are not microbes. They are large, conspicuous, well-studied animals that have been appearing in scientific papers, aquarium displays, and nature documentaries for nearly a century and a half. And we still could not tell them apart. The species problem is connected to a broader issue in deep-sea biology. We study what we can reach, we classify what we can see, and when our tools are limited, our conclusions are limited too. For the giant isopod, the limitation was particularly insidious, because the animal looked similar enough across species that nobody questioned whether they were dealing with a single species or several. The assumption was baked in from the beginning. Milne Edwards described Baphonemus giganteus in 1879. For the next 137 years, every large isopod pulled from the Atlantic was assumed to be the same thing. It took molecular genetics to reveal the truth, and molecular genetics only became widely available and affordable in the last few decades, which means that for the vast majority of the time we have been studying giant isopods, we simply did not have the tools to see what was right in front of us. This should give us pause. Not just about isopods, but about every assumption we have built on limited data, from limited tools in the largest and least explored habitat on the planet. The deep ocean covers more of the Earth's surface than all the continents combined. We have explored less than 20% of it, and the organisms we have studied from that 20%, we have often studied from dead specimens hauled to the surface in nets. The giant isopod is a cautionary tale about the distance between what we think we know and what is actually true. But if the species question was humbling, the question of reproduction is something else entirely. It is one of the deepest, most persistent mysteries in deep-sea biology. And in the case of the giant isopod, it borders on the eerie. Giant isopod reproduction is one of the least understood aspects of their biology. And what we do know is equal parts fascinating and unsettling. Here is what we know for certain. Mature female giant isopods develop a structure called a marsupium, a brood pouch formed by overlapping plates that grow from the bases of their legs. It is not unlike the pouch of a kangaroo in principle, though it looks nothing like one. The marsupium is a ventral enclosure, a chamber on the underside of the body, protected by the overlapping plates. Inside this pouch the female carries her eggs. These eggs are remarkable. They are thought to be among the largest eggs produced by any marine invertebrate. Each one is roughly the size of a large marble. and when the young hatch, they do not emerge as larvae. There is no planktonic stage, no drifting through the water column as tiny vulnerable specks of life. Instead, the young emerge as fully formed miniature adults. They are called mankai, and they are already recognizable as isopods when they leave the pouch. They have legs, antennae, eyes, armor plating. The only thing they lack is their 7th pair of legs, which develops later. At birth they are approximately 60 millimeters long, about the length of a human finger, fully formed, fully functional, ready for life on the seafloor. This reproductive strategy is called direct development, and it is relatively unusual in the marine world. Most marine invertebrates produce enormous numbers of tiny eggs that hatch into larvae and drift through the water, with the understanding that the vast majority will be eaten before they ever reach adulthood. It is a numbers game. The more offspring you produce, the better the odds that a few will survive. Giant isopods take the opposite approach. They produce relatively few eggs, but each egg is large and well provisioned. Each offspring is born large enough and developed enough to survive from the moment it enters the world. Quality over quantity. A strategy that makes sense in an environment where the risks of being small and vulnerable in open water are extreme. But here is where the mystery begins. Despite over 140 years of study, despite the examination of thousands of specimens pulled from the deep ocean, we have almost no observational data on giant isopod reproduction. And the reason for this is startling. As of the most recent comprehensive reviews, only 2 egg-bearing female giant isopods had ever been documented in the wild. 2. In 140 years. Think about that for a moment. Scientists have been collecting giant isopods since 1879. They have pulled them from the trawl nets, baited traps, and submersible collection devices across the Atlantic, Pacific, and Indian Oceans. They have examined thousands of individuals. They have dissected them, measured them, sequenced their DNA, published hundreds of papers about them. And in all of that time, all of that effort across all of those specimens, they have found precisely 2 females carrying eggs. Where are the mothers? The leading hypothesis is both elegant and deeply strange. Scientists believe that brooding female giant isopods bury themselves in the sediment of the ocean floor and stop foraging entirely. They go underground. They disappear. They essentially remove themselves from the active population for the entire duration of the brooding period, which is believed to last many months. This makes sense from a survival standpoint, even though it sounds extreme. A female carrying a pouch full of large, nutrient-rich eggs is vulnerable in ways that a non-brooding isopod is not. The marsupium restricts her mobility. The eggs add weight and bulk. She cannot curl into a defensive ball as effectively because the pouch is on her underside. And in the deep ocean, where every meal is an opportunity and every encounter with a larger animal is a potential death sentence, being slow, heavy, and unable to defend yourself properly is a serious liability. So the brooding female digs into the soft sediment of the seafloor and waits. She does not eat. She conserves energy using her extraordinarily slow metabolism, the same adaptation that allows non-brooding isopods to survive years without food. She incubates her eggs in hiding, protected from predators and scavengers by the sediment itself, and she stays there, invisible to nets and traps and submersibles, until the eggs develop and the young are ready to emerge. This is why we almost never find them. They are not in the water column. They are not on the surface of the seafloor. They are buried in it, in sediment that trawl nets pass over and baited traps cannot reach. They have, in effect, evolved a reproductive strategy that makes them invisible to every method of collection we commonly use. We do know a few additional things from the limited data we have. Reproduction appears to peak during autumn and winter, based on the seasonal abundance of juvenile specimens in the trawl catches. The timing makes sense. Autumn and winter in the deep sea correspond to periods when surface productivity, and therefore the rain of organic material to the bottom, is often at its highest. Young isopods emerging during a period of relatively greater food availability would have a better chance of survival. We also know that a brooding female faces a dangerous trade-off. If she eats too much before brooding, her stomach expands to fill 2/3 of her body cavity, which could compress or damage the developing eggs in the marsupium. So she must balance energy storage against reproductive safety. She needs enough fat reserves to survive months without eating, but not so much food in her stomach that it threatens the brood. This is a fine line, and it is one that each female must navigate on her own, in the dark, at the bottom of the ocean. There is also evidence that larger, sexually mature individuals tend to be found in shallower water within the giant isopods range. This has led some researchers to speculate that females may migrate to slightly shallower depths to brood, where the sediment might be softer or the temperature slightly different, but this remains speculative. We simply do not have enough data to confirm it, and that is the honest summary of what we know about giant isopod reproduction. Almost nothing. We know the anatomy, we know their eggs are large, we know the young are born fully formed, we know the mothers vanish. Beyond that, nearly everything is inference and educated guessing. This should be disturbing in a scientific sense. The giant isopod is not an obscure animal. It is one of the most frequently cited examples of deep-sea life. It appears in museum exhibits around the world. It has its own Wikipedia page with dozens of cited sources. It has been featured in countless documentaries. And yet, when it comes to one of the most fundamental aspects of its biology, how it reproduces, we are essentially in the dark. The reproductive mystery connects to a broader theme in the giant isopod story, which is the sheer depth of our ignorance about the deep ocean. We know more about the surface of Mars than we do about the bottom of our own ocean. That is not an exaggeration. It is a frequently cited observation in marine science, and it remains true despite decades of technological advancement. The deep sea is difficult to reach, difficult to study, and expensive to explore. Every dive costs thousands or tens of thousands of dollars. Every expedition requires specialized equipment, trained crews, and significant logistical support, and the deep sea is vast. It covers over 60% of the Earth's surface. We have explored less than 20% of the ocean floor with any level of detail. So when we say we were wrong about the giant isopod, we should be careful to note that the error was not purely scientific. It was logistical. We were wrong because we could not visit. We were wrong because our tools were limited. We were wrong because the deep ocean is, even now, one of the most difficult environments on Earth to study. And the giant isopod, patient and still in the darkness, waited 140 years for us to develop the technology to see it clearly. But the isopod's mysteries do not end with reproduction. They extend backward in time, hundreds of millions of years, to a lineage so ancient that it predates the dinosaurs, the flowering plants, and the breakup of the supercontinent Pangaea. And they extend forward in time to a set of emerging threats that may, for the first time in its immense history, put the giant isopod's survival at risk. The giant isopod is what biologists sometimes call a living fossil, though that term is somewhat misleading. It does not mean the animal has not evolved. It has. The genome study made that abundantly clear. The giant isopod's DNA has been actively changing and adapting for hundreds of millions of years, driven by those 84% transposable elements that give its genome such remarkable plasticity. What living fossil means in this context is that the basic body plan, the external appearance, the fundamental shape of the animal, has remained remarkably consistent across an almost incomprehensible span of time. Fossil records show that the genus Bathynomus dates back at least 160 million years. That places the earliest known giant isopods in the Jurassic period. When these animals first appeared, Pangaea was still in the process of breaking apart. The Atlantic Ocean did not yet exist in its current form. Dinosaurs dominated every terrestrial ecosystem on the planet. Flowering plants had not yet evolved. The earliest known birds were just beginning to take to the air, and giant isopods were already there, trundling across the dark ocean floor in bodies that looked almost exactly like the bodies they wear today. They survived the extinction event that ended the Cretaceous period 66 million years ago, the asteroid impact that wiped out the non-avian dinosaurs and roughly 75% of all species on Earth. They survived it because the deep ocean is a buffer against catastrophe. When the surface world burns, when dust clouds block the sunlight and temperatures plummet and food chains collapse from the top down, the deep ocean persists. Its energy sources are different. Its food web is largely decoupled from sunlight. The organisms that depend on chemosynthesis, on the chemical energy that seeps from the Earth's interior, can continue even when the surface goes dark. And the organisms that depend on the slow rain of organic matter from above, like the giant isopod, can survive extended periods of reduced food supply because they are already adapted to famine. The giant isopod survived every ice age, every period of volcanic activity, every shift in ocean chemistry, every mass extinction event. It has been doing essentially the same thing, in essentially the same way, in essentially the same environment, for longer than most lineages of mammals have existed, and specimens collected from different parts of the world, from the Gulf of Mexico, from the waters off Australia, from the Indian Ocean, despite being different species, are almost identical in appearance. The deep sea rewards consistency. When your environment does not change much, neither do you, but the environment is changing now, and the changes are coming from above. Analysis of the stomach contents of giant isopods collected in the southern Gulf of Mexico found large quantities of plastic, not microplastics alone, though those were present too, larger fragments of synthetic material, debris that has sunk from the surface world into the deep, infiltrating an ecosystem that existed for hundreds of millions of years without ever encountering anything like polyethylene or polypropylene. This is not unique to giant isopods. Plastic contamination has been found in organisms at every depth of the ocean, from surface-dwelling fish to amphipods living in the Mariana Trench, the deepest point on Earth. But the finding is particularly stark in the context of the giant isopod's biology. Remember the stomach. 2/3 of the body cavity, genetically optimized for maximum extraction. Giant isopods are built to eat everything they can and to squeeze every possible calorie from every meal. They do not have the biological machinery to differentiate between organic food and synthetic debris. They eat what is available, and increasingly what is available includes plastic. The effects of plastic ingestion on giant isopods are not well studied. In other deep-sea organisms, plastic contamination has been linked to reduced reproductive success, disrupted endocrine function, and physical damage to the digestive tract. Whether the same effects occur in giant isopods is unknown, but given that these animals already reproduce slowly, with small clutches of large eggs and extended brooding periods during which the mother does not eat, any additional stress on the reproductive system could have outsized effects on population stability. Deep-sea trawling is another growing concern. Commercial trawling operations that drag heavy nets across the ocean floor can destroy the habitat that giant isopods depend on, and can capture brooding females who, as we discussed, are already so rare in the observable population that we have documented only 2 in over a century. The loss of even a few reproductive females from a slow reproducing species can have cascading effects on local populations, and because we know so little about giant isopod population dynamics, we may not even recognize the damage until it has already become severe. Climate change may pose the most fundamental threat of all. Giant isopods are cold water animals. Research on Bathynomus giganteus has shown that the species is most abundant in water temperatures between 3 and 13 degrees Celsius. That is a narrow range by mammalian standards. As ocean temperatures rise, driven by the absorption of excess atmospheric heat, the habitable zone for these animals narrows. The deep ocean is not immune to warming. It responds more slowly than the surface, but it does respond. And for animals that have spent 160 million years in a cold, stable environment, Even a modest shift in the temperature could push them beyond the boundaries of their tolerance. Some researchers have speculated that deep-sea gigantism itself may be vulnerable to ocean warming. The phenomenon depends on a delicate balance of temperature, dissolved oxygen, metabolic rate, and energy efficiency. If the water warms, metabolic rates increase. If metabolic rates increase, energy reserves are burned faster. If energy reserves are burned faster, the advantage of being large in a food-scarce environment diminishes. The very thing that makes the giant isopod what it is, its size, could become a liability in a warmer ocean. This is speculative. We do not have long-term population data for giant isopods. We do not know their current population sizes with any confidence. We do not know how quickly they can adapt to changing conditions, though the extraordinary plasticity of their genome suggests they may be more flexible than their static body plan implies. But the risks are real, and they are growing, and they are the kind of risks that we tend to ignore until it is too late, because the animals affected live in a place we almost never visit. There is something worth pausing on here, something that the genome study, the species discovery, the behavioural footage, and the reproductive mystery all point toward, but that we have not yet said directly. We do not understand the deep ocean. Not really. Not in the way we understand forests or grasslands or coral reefs, or even the surface waters of the open sea. The deep ocean is the largest habitat on Earth, and it is the one we know the least about. And the giant isopod, this animal we thought we had figured out, is a vivid illustration of exactly how wide the gap between our knowledge and reality actually is. Consider the numbers for a moment. The deep sea, defined as ocean below 200 meters, covers approximately 361 million square kilometers. That is more than the total land surface of the Earth. It is more than twice the area of all the continents combined. And within that vast expanse, we have explored at best a fraction. Estimates vary, but the most commonly cited figure is that we have mapped only about 23% of the ocean floor with modern multi-beam sonar, and we have directly observed with submersibles, remotely operated vehicles, or deep-sea cameras a much smaller percentage than that. The number of locations where we have actually collected biological samples from the deep-sea floor is smaller still. This means that when we describe the ecology of the deep ocean, we are extrapolating from an extremely limited data set. We are making claims about the largest ecosystem on the planet based on what amounts to a handful of samples taken from a handful of locations. It is like trying to describe the ecology of an entire continent based on observations from a few city parks. The giant isopod lived comfortably within this gap in our knowledge for over a century. It was well known enough to appear in the textbooks, but poorly understood enough that fundamental errors in our understanding could persist for generations. And it is not alone. The deep ocean is full of organisms that we have named and classified based on dead specimens and fragmentary observations, organisms whose behaviour, ecology, and evolutionary history may be just as misunderstood as the giant isopods was. Let me give you another dimension of the isopod that deserves more attention than it typically receives. Its eyes. Giant isopods have compound eyes, not the simple eyes that many deep-sea creatures have reduced or eliminated entirely. Compound eyes, the same basic eye type found in insects, crabs, and other arthropods. But the giant isopod's compound eyes are extraordinary even by compound eye standards. Each eye contains nearly 4,000 individual facets, called ommatidia. Each facet is a tiny optical unit that collects light from a slightly different angle, and the brain combines the inputs from all 4,000 facets into a single composite image. This is essentially the same principle that modern digital cameras use when they combine information from millions of individual pixels. But here is what makes the giant isopod's eyes particularly remarkable. They are backed by a reflective layer called the tapetum lucidum. You have encountered this structure before, even if you did not know its name. It is the structure that makes a cat's eyes glow when light hits them in the dark. It is the structure that gives alligators their eerily reflective eyes at night. And it is the structure that gives giant isopods the ability to see in conditions of near total darkness. The tapetum works by reflecting light that has already passed through the photoreceptor cells back through them a second time. This effectively doubles the amount of light available for the retina to process. In terrestrial animals, this adaptation is associated with nocturnal hunting. Cats, wolves, deer, and raccoons all have tapetum because they need to see in low light. In the deep ocean, where sunlight does not penetrate below about 1,000 meters, the giant isopod's tapetum serves the same function. It allows the animal to detect the faintest traces of bioluminescence, the tiny flashes of biological light produced by other deep-sea organisms. It allows it to navigate in conditions where a human eye would see nothing at all. The fact that the giant isopod has invested so heavily in vision is itself a clue about its lifestyle. Vision is metabolically expensive. Maintaining nearly 4,000 ommatidia per eye, plus the reflective tapetum, requires significant biological resources. If the giant isopod were truly a passive scavenger that simply sat in one place and waited for food to land on it, there would be no evolutionary advantage to such sophisticated eyes. The fact that it has them suggests that it actively uses vision to navigate its environment, to detect food from a distance, to identify potential threats, and possibly to identify potential mates. It suggests an animal that is far more aware of its surroundings and far more active in responding to them than the scavenger narrative would have us believe. The eyes also connect to an interesting question about deep-sea gigantism. In many deep-sea species, the trend toward larger body size is accompanied by a reduction or loss of visual structures. The rationale is simple. If you live in permanent darkness, why invest energy in eyes? Many deep-sea fish have reduced eyes or no eyes at all. Some deep-sea crustaceans have similarly reduced visual systems. The giant isopod breaks this pattern completely. It is one of the largest deep-sea invertebrates, and it has some of the most sophisticated eyes in the deep ocean. This combination of gigantism and visual investment is unusual and, as far as I am aware, not fully explained by any current theory. One possibility is that giant isopods occupy a depth range where some residual light still exists. The majority of specimens have been collected between approximately 170 meters and 2,140 meters deep. At the shallower end of this range, there may be trace amounts of downwelling sunlight, not enough to illuminate the environment in any meaningful way, but perhaps enough to provide a faint gradient that a highly sensitive eye could detect. At the deeper end, bioluminescence is the only light source. In either case, having eyes that can detect extremely faint light confers an advantage that evolution has clearly rewarded. But there is another possibility, less commonly discussed but worth considering. The eyes may serve a social function. We know almost nothing about giant isopod social behavior. We do not know how males and females find each other in the vast darkness of the deep ocean. We do not know whether giant isopods aggregate in groups, whether they maintain territories, whether they communicate in any way. But the fact that they have such well-developed eyes raises the possibility that visual signals play some role in their interactions with one another. Perhaps they can detect the reflected glow of each other's tapetum. Perhaps bioluminescent organisms in their environment serve as landmarks or navigation aids. Perhaps there are behaviours occurring in the deep ocean between living giant isopods that we have simply never observed because we have never been able to watch them for long enough or in the right conditions. This is one of the most frustrating aspects of deep-sea biology. We can speculate endlessly based on anatomy and genetics, but until we can observe these animals in their natural environment, undisturbed, over extended periods, our understanding will always be incomplete. And the technology to do this, long-duration deep-sea observation platforms with high-resolution cameras and minimal environmental disturbance, is still in its infancy. Let me take you deeper into the anatomy, because the more you learn about the giant isopod's body, the more you realise how precisely engineered it is for its environment. We have already discussed the stomach, which can expand to fill 2/3 of the body cavity, and we have discussed the fat body, which stores lipids so efficiently that the animal can survive years without food. But there are other features worth examining. The exoskeleton first. Giant isopods have a thick, calcareous shell composed of overlapping segments, similar in principle to the armour of medieval knights. The shell is divided into a head shield, 7 thoracic segments, and a series of abdominal segments that end in a broad tail section called the pleotelson. Each segment overlaps the one behind it, creating a continuous armour surface that can flex with the animal's movements while still providing protection. The shell serves multiple purposes. It protects against predators, though as we noted the giant isopod has few natural enemies. It resists the immense hydrostatic pressure of the deep ocean. Though the animal's internal fluids are already at equilibrium with the surrounding water, so pressure resistance is less about structural strength and more about maintaining the integrity of the body plan, and it provides a framework for the attachment of muscles, particularly the powerful muscles that operate the 4 sets of jaws and the 7 pairs of walking legs. When threatened, the giant isopod can curl into a ball, tucking its legs and soft ventral surface inside the dome of armoured plates. This behaviour is identical to what terrestrial woodlice do when disturbed, and it is one of the clearest visible links between the deep-sea giant and its garden-dwelling relatives. The defensive ball exposes only the hard dorsal shell to potential attackers. It is simple, effective, and it has apparently worked for at least 160 million years. The 7 pairs of thoracic legs deserve attention as well. The first pair is modified into maxillipeds, which are essentially feeding appendages. They manipulate food and bring it to the 4 sets of jaws. The remaining 6 pairs are walking legs, used for locomotion on the seafloor. Giant isopods are not fast walkers. They move with a measured, deliberate gait, placing each leg carefully. This is consistent with an animal that conserves energy obsessively, for which every unnecessary movement represents a waste of precious metabolic resources. The abdominal section, or pleon, has 5 segments, each bearing a pair of biramous pleopods. These are flattened paddle-like appendages with 2 branches. The branches serve dual functions. They can be used for swimming, providing a slow but sustained propulsion through the water, and the flat membranous surfaces of the pleopods function as gills, exchanging oxygen and carbon dioxide with the surrounding water. This is a remarkably efficient design. The same structures that move the animal also breathe for it. Giant isopods have 2 pairs of antennae, and in some species the first pair can extend to nearly half the body length. These antennae are sensory organs, densely packed with chemoreceptors and mechanoreceptors. In the permanent darkness of the deep ocean, chemical sensing is arguably more important than vision. The antennae allow the isopod to detect trace chemicals dissolved in the water, chemicals released by decomposing organic matter, by living organisms, by the sediment itself. An isopod sitting on the seafloor with its antennae extended is not passively waiting. It is actively sampling the chemical environment around it, building a map of what is nearby based on dissolved molecules rather than light. This chemical sensing ability helps explain how giant isopods find food in such a vast, dark environment. When a whale carcass settles to the bottom, it releases a plume of chemical signals as it begins to decompose. These signals travel through the water, spreading outward from the carcass in all directions. An isopod may be kilometers away, but if the chemical plume reaches its antennae, it can orient toward the source and begin walking. This is how baited traps work. Scientists lower containers of fish or other organic material to the seafloor, and within hours, giant isopods begin to arrive, drawn by the chemical trail. Some of the largest congregations of giant isopods ever observed have been at baited camera stations, where dozens of individuals converge on a single food source, having navigated through the darkness using nothing but their sense of smell. The convergence behaviour at food sources is itself interesting. Giant isopods are not typically considered social animals, and in most of the footage we have, they appear to operate independently. But at whale falls and baited stations, they aggregate in large numbers and feed communally. There is no obvious aggression between individuals during these feeding events, which is unusual for animals competing for a scarce resource. This suggests either that the food source is large enough to reduce competition to negligible levels, which is likely the case with the whale falls, or that giant isopods have some mechanism for tolerating the presence of conspecifics during feeding that we do not yet understand. The feeding behaviour itself is fascinating to watch if you can find the footage. When a giant isopod arrives at a food source, it does not delicately sample the material. It gorges. It uses its 4 sets of jaws to tear through flesh, shell, bone, whatever is available. It eats rapidly and voraciously, distending its body as the stomach expands to accommodate as much food as possible. Some observers have described post-feeding isopods as so bloated that they can barely move, their bodies swollen to grotesque proportions, their legs struggling to support the added weight. This gorging behaviour makes perfect sense in the context of their metabolism. When you never know when your next meal will arrive, the rational strategy is to eat as much as you possibly can every time you get the chance. Store it all. Burn it slowly. Wait for the next opportunity. The genome confirms this. The expanded glycolysis genes in the digestive organs ensure maximum caloric extraction. The suppressed lipid degradation genes ensure the stored fat lasts as long as possible. The enormous stomach ensures maximum intake. Everything in the giant isopod's biology points toward a single overriding imperative. When food is available, eat everything, and when food is not available, survive on what you have stored. The 5-year fasting record set by the specimen at the Toba Aquarium in Japan is almost certainly not the upper limit of the giant isopod's fasting ability. That specimen was in captivity, in an aquarium where conditions were controlled and where the animal may have been experiencing stress from captivity itself. In the wild, where conditions are optimized by hundreds of millions of years of evolution, the true fasting capacity may be even longer. We simply do not know. Consider what that means from a human perspective. 5 years without food. Imagine not eating for 5 years. Not a day. Not a week. 5 years. For a human, that is approximately 1,826 days. For the giant isopod, it is apparently survivable if not comfortable. The metabolism slows to a crawl. The fat reserves are drawn down fraction by fraction. The animal enters a state of profound energy conservation, reducing its activity to the absolute minimum required to stay alive. Breathing, sensing, waiting. It is tempting to anthropomorphize this, to imagine what it would feel like to sit in the dark on the bottom of the ocean, not eating, not moving for 5 years. But the giant isopod is not experiencing this the way a human would. Its nervous system is not built for the kind of such subjective suffering that prolonged hunger causes in mammals. It is simply in a state of metabolic holding, burning the absolute minimum energy required to maintain basic biological function, waiting with the infinite patience of an organism that has been playing this game for 160 million years for the next meal to arrive. And when that meal arrives, it will eat everything. Let me widen the frame for a moment. Because the giant isopod does not exist in isolation. It is part of an ecosystem, one of the strangest and most poorly understood ecosystems on the planet. And understanding the isopod means understanding, at least in broad strokes, the world it inhabits. The deep-sea floor is not the barren wasteland that early scientists imagined. It is not the Azoic desert that prevailed in scientific thinking before the Challenger expedition of the 1870s, but it is also not a bustling, teeming habitat like a coral reef or a kelp forest. The deep-sea floor is something in between. It is sparse, it is slow, it is immensely old, and it operates on timescales that would make surface ecosystems seem frantic by comparison. The primary energy input for most of the deep-sea floor is what marine biologists call marine snow. This is the constant slow drift of organic particles from the sunlit surface waters to the depths below. Dead phytoplankton, fecal pellets from zooplankton, fragments of dead animals, mucus webs discarded by gelatinous organisms. All of this material sinks in slowly, through thousands of meters of water, and a fraction of it eventually reaches the bottom. By the time it arrives, it has been partially consumed and partially decomposed by organisms in the water column above. What reaches the sea floor is a thin, irregular dusting of organic material, enough to sustain life, but not enough to sustain much of it. This is the fundamental constraint of life in the deep ocean. Energy is scarce, calories are precious, and every organism on the deep sea floor has evolved strategies to deal with this scarcity. Some, like the giant isopod, eat rarely but massively, storing energy in fat reserves that can last years. Others, like deep-sea sea cucumbers, process vast quantities of sediment, extracting whatever trace nutrients they can find in the mud. Still others, like the organisms living near hydrothermal vents and cold seeps, tap into chemical energy from the Earth's interior, using chemosynthesis instead of photosynthesis to build organic molecules from inorganic raw materials. The giant isopod occupies a specific niche within this ecosystem. It is a mobile, opportunistic consumer that exploits the patchily distributed food resources of the deep floor. Whale falls are the bonanza events, the jackpots. A single dead whale can sustain a local community of organisms for decades, and giant isopods are among the organisms that benefit. But whale falls are rare and unpredictable. Between them, the isopod must survive on whatever it can find: dead fish, dead squid, living sea cucumbers, the occasional dogfish shark that makes the mistake of getting too close. The distribution of giant isopods across the world's oceans is itself informative. They are found in the Atlantic, Pacific, and Indian Oceans, but no single species spans more than one ocean basin. The four known Atlantic species are B. obtusus, B. miare, B. maxiorum, and B. giganteus, with the newly described B. yucatanensis now bringing the total in the Gulf of Mexico to 3. The remaining species are distributed across the Indo-Pacific. No species is known from both the Atlantic and the Indo-Pacific. This distribution pattern tells us something about the evolutionary history of the genus. When the supercontinent Pangaea began to break apart starting roughly 200 million years ago, it split the ancient oceans into the modern Atlantic and Pacific basins. Populations of isopods that had once been connected were separated by the growing land masses. Over tens of millions of years, isolated from each other, these populations diverged into distinct species. The Atlantic species and the Indo-Pacific species share a common ancestor, but have been evolving independently for an immense period of time. This is the same pattern seen in many deep-sea organisms. The deep ocean, despite being connected through bottom currents and water masses, is effectively divided into isolated basins by continental land masses, underwater ridges, and differences in temperature and chemistry. Organisms that live on the deep floor, unable to cross these barriers, speciate in isolation. The result is a patchwork of closely related but distinct species across the world's ocean basins, each one adapted to the specific conditions of its local environment. For the giant isopod, this biogeographic pattern has an additional implication. It means that the roughly 20 known species probably represent an undercount. If different species look so similar that they could fool trained biologists for over a century, then it is virtually certain that additional cryptic species exist, species that are morphologically indistinguishable from known species, but genetically distinct. Only molecular genetics can reveal these, and molecular genetics requires fresh tissue samples, which in turn requires new collecting expeditions to poorly sampled regions. It is a slow, expensive process, but based on what we have seen so far, it is likely to yield surprises. Now I want to bring the conversation back to something we touched on earlier and explore it more deeply: the relationship between the giant isopod and its terrestrial relatives, the common woodlouse. Because this relationship is more than a fun fact. It is a window into one of the great stories of evolutionary biology. Isopods as a group are ancient. The order Isopoda dates back to at least the Carboniferous period, over 300 million years ago. The earliest isopods were marine, living in the oceans long before any animal had colonized the land. Over evolutionary time, some lineages of isopods made the transition from sea to land, evolving the ability to breathe air, to conserve water, and to survive in terrestrial environments. These are the woodlice, the pillbugs, the roly-polies that live under logs and in garden compost heaps around the world. The phylogenetic analysis from the 2022 genome study estimated that the deep-sea isopod lineage and the terrestrial isopod lineage diverged approximately 245 million years ago during the early Triassic period. This means that the common woodlouse in your garden and the giant isopod at the bottom of the ocean share a common ancestor from roughly the same time that the first dinosaurs were appearing on Earth. Think about what that means. For 245 million years, these two lineages have been evolving in completely different environments. One adapted to life on land, developing the ability to breathe air through modified gill-like structures, to roll into a defensive ball to prevent water loss, and to thrive in the damp sheltered microhabitats of the terrestrial world. The other adapted to life in the deep ocean, developing enormous body size, extreme metabolic efficiency, massive energy storage capacity, and the ability to survive years without food in total darkness under crushing pressure. And yet, despite 245 million years of separate evolution, they still look remarkably similar. The basic body plan, the segmented armour, the multiple pairs of legs, the ability to curl into a ball, is shared between the garden woodlouse and the deep-sea giant. This is a testament to how successful the isopod body plan is. It works on land and it works in the deep sea. It works at 1 centimetre and it works at 76 centimetres. It has persisted across geological epochs, through mass extinctions, through radical changes in climate and ocean chemistry. The isopod body plan is, in a very real sense, one of the most successful designs in the history of life on Earth. This brings up an interesting philosophical question that I have been turning over in my mind while researching this topic. What does it mean to be successful, biologically speaking? We tend to measure biological success in human terms: intelligence, complexity, social behaviour, tool use. We look at the natural world and implicitly rank organisms by how much they resemble us. Mammals are more successful than reptiles. Primates are more successful than rodents. Humans are the pinnacle. This is a bias so deep that most people do not even recognize it as a bias, but by almost every objective measure of biological success, the giant isopod is more successful than humans. It has existed for at least 160 million years. Homo sapiens has existed for roughly 300,000 years. The isopod has survived every mass extinction event of the last 160 million years. Humans have not yet been tested by a single one. The isopod's body plan has been stable and functional across geological time. Our bodies are the product of relatively recent and rapid evolution, still riddled with design compromises, back pain, wisdom teeth, an immune system that sometimes attacks itself, that reflect the haste of our evolutionary journey. The isopod has colonized the deep ocean, an environment that covers more of the Earth's surface than all terrestrial habitats combined. We cannot even visit the deep ocean without elaborate technological support. The isopod can survive 5 years without eating. A human would be dead in a few weeks. The isopod's genome, nearly twice the size of ours, is packed with adaptive flexibility. Our genome, for all its achievements, is relatively compact. None of this means the isopod is better than a human in any meaningful sense, but it does mean that our definition of biological success is narrow and self-serving. The giant isopod is a master of its environment. It has been a master of its environment for longer than there have been flowers on Earth, and the fact that it achieves this mastery through stillness, patience, and metabolic efficiency rather than through intelligence, speed, or social complexity does not make it any less remarkable. In the deep ocean, survival is not about being the fastest or the smartest. It is about being the most efficient, the most patient, the most perfectly adapted to an environment that has barely changed in 100 million years. And by that measure, the giant isopod is among the most successful animals that has ever lived. Let me return to the physical environment for a moment, because I think it is worth spending some time on what it actually means to live where the giant isopod lives. The depth range for giant isopods is approximately 170 meters to 2,140 meters, though some records suggest they may occur somewhat deeper. This is a vast vertical range, spanning from the edge of the continental shelf to the abyssal plain. At the shallower end there may be faint traces of sunlight. At the deeper end the only light comes from bioluminescence. The pressure at 1,000 meters deep, roughly the middle of the giant isopod's range, is approximately 100 atmospheres. That means the water pressure is 100 times greater than the atmospheric pressure at sea level. At 2,000 meters, it is 200 atmospheres. A human body placed at these depths without protection would be crushed. The lungs would collapse. The sinuses would implode. Every gas-filled space in the body would be compressed to a fraction of its normal volume. But the giant isopod has no gas-filled spaces. Its body is essentially incompressible. Its tissues are saturated with water at the same pressure as the surrounding environment. Pressure, for the isopod, is not a crushing force. It is simply a fact of life, as unremarkable and constant as gravity is for us. The temperature at these depths is cold, between 3 and 13 degrees Celsius for the majority of the giant isopod's range, though this varies by location and depth. This is roughly the temperature of a refrigerator. The giant isopod's entire life unfolds at refrigerator temperature in total darkness, under pressures that would kill a human in seconds. And it's been doing this for 160 million years. The water at these depths is still. There are currents, but they are slow and gentle compared to the turbulent surface ocean. The energy of waves does not reach the deep floor. The seasonal storms that churn the surface waters dissipate long before they affect the abyss. The deep ocean is, in many ways, the most physically stable environment on Earth. Temperature barely changes. Pressure is constant. Light never varies because there is never any light. The only thing that changes, the only variable in an otherwise unchanging world, is the occasional arrival of food. This stability is the key to understanding why the giant isopod and many other deep-sea organisms have changed so little over geological time. Evolution is driven by environmental change. When conditions shift, organisms must adapt or die. When conditions remain constant, there is little pressure to change. The deep ocean has been cold, dark, and high pressure for hundreds of millions of years. The organisms that live there have had no reason to dramatically alter their body plans because the body plans they have work perfectly well in an environment that has not significantly changed. This is not the same as saying these organisms have not evolved. They have. The genome study proved that. The giant isopod's DNA has been actively changing, driven by transposable elements and the slow accumulation of beneficial mutations. But the changes have been internal, invisible to the eye. The external body plan, the armoured segments, the compound eyes, the 7 pairs of legs has remained stable because external change was not required. The deep ocean did not demand a new design. It demanded refinement of an existing one. There is something almost meditative about this idea. An animal that has spent 160 million years refining a single design for a single environment. No dramatic transformations, no branching into radically different forms, just the slow, patient, invisible optimization of an architecture that already worked. while the surface world was convulsing through mass extinctions, continental collisions, ice ages, and the explosive diversification of mammals, birds, and flowering plants. The giant isopod was on the bottom of the ocean, in the dark, in the cold, doing essentially the same thing it had always done, eating when it could, waiting when it could not, surviving. I want to talk for a moment about what happens when a giant isopod is brought to the surface and placed in an aquarium, because the aquarium stories have become some of the most famous anecdotes about this animal, and they reveal something important about how the public relates to deep-sea life. The most well-known aquarium isopod story comes from the Toba Aquarium in Mie Prefecture, Japan. In 2007, the aquarium acquired a giant isopod, a specimen they identified as Baphinomus giganteus, and placed it in a deep-sea exhibit. For the first 2 years, the animal ate normally. It consumed horse mackerel offered by its keepers and seemed to settle into captive life without obvious difficulty. Then, in January 2009, it stopped eating. It refused horse mackerel. It refused squid. It refused every food item the keepers offered. Days turned into weeks. Weeks turned into months. Months turned into years. The keepers were baffled. The animal showed no obvious signs of illness. It moved around its tank. It responded to stimuli. It appeared by all external measures to be healthy. It simply would not eat. The isopod's hunger strike, as it was quickly dubbed by the Japanese media, became a national sensation. The animal was given the designation number 1 and became arguably the most famous individual invertebrate in Japan. News outlets tracked its fasting streak the way they might track a sports record. Social media users posted updates. The aquarium reported that visits increased specifically because people wanted to see the isopod that refused to eat. Number 1 fasted for 5 years and 43 days. It finally died on February 14th, 2014. Valentine's Day. The cause of death was not determined with certainty, but an autopsy revealed no signs of disease. The animal simply appeared to have exhausted its reserves. The story of Number 1 is often told as a curiosity, a quirky tale about a strange deep-sea animal doing something inexplicable. But I think it reveals something deeper. It reveals how the giant isopod's biology, its extreme metabolic efficiency, its capacity for prolonged fasting, its patience, appears to us as alien. We cannot relate to an animal that does not eat for 5 years. It violates our deepest intuitions about what it means to be alive. Living things eat. Living things need energy. An animal that refuses food for 5 years seems defective, or stubborn, or impossible. We search for explanations in human terms. Was it depressed? Was it sick? Was it protesting? None of these apply. The isopod was simply doing what its biology allows it to do. Waiting. But the aquarium stories also highlight a genuine scientific problem. We do not know why Number 1 stopped eating. We can speculate that the captive environment did not provide the right cues. Perhaps the food offered did not trigger the animal's feeding response. Perhaps the temperature, the pressure, the lighting, or some other environmental variable was subtly wrong. Perhaps the isopod had eaten enough in its first 2 years to sustain itself and simply entered a fasting state that, in the wild, would have ended whenever the next food source appeared. In captivity, with food being offered on a human schedule, the isopod may not have recognized the offered food as a trigger to resume feeding. This is a broader problem with keeping deep-sea animals in captivity. The conditions of the deep ocean are extraordinarily difficult to replicate. The pressure alone is beyond what most aquarium systems can maintain. Most deep-sea animals kept in captivity are housed at surface pressure, which is a fraction of what they experience in the wild. The long-term effects of this pressure, difference on their behavior, physiology, and health are poorly understood. We are essentially asking animals that have spent their entire evolutionary history under 100 to 200 atmospheres of pressure to live at 1 atmosphere. It is the equivalent of asking a human to live at the summit of Mount Everest permanently. except the differential is far more extreme. Despite these challenges, giant isopods have become increasingly popular in public aquariums, particularly in Japan, where they have achieved a kind of cult status. Plush toys shaped like giant isopods are sold in museum gift shops. Giant isopod-shaped phone cases, keychains, and even bread exist. There is a giant isopod-themed restaurant in Taiwan, where the decor features oversized isopod sculptures and the menu includes isopod-shaped dishes. This cultural embrace of the giant isopod is, in a way, encouraging. It suggests that the public is fascinated by deep-sea life, willing to engage with organisms that are genuinely strange and alien-looking, and capable of developing affection for animals that bear no resemblance to the charismatic megafauna. The pandas, the dolphins, the elephants, that typically dominate conservation messaging. The giant isopod is not cute in any conventional sense. It is armoured, multi-legged, and vaguely menacing, but people love it. They buy plush toys of it. They follow its aquarium exploits on social media. They care about it. Whether this cultural affection translates into meaningful conservation attention is another question entirely. As we discuss, giant isopods face real threats from plastic pollution, deep-sea trawling, and climate change. But these threats are invisible to most people because they occur in a habitat that most people will never see. The deep ocean does not generate the same emotional urgency as a burning forest or a bleached coral reef. It is out of sight, and for most of the world's population, out of mind. I think this is one of the most important challenges in marine conservation today. How do you make people care about an ecosystem they cannot visit, filled with organisms they will never see in person, facing threats that are abstract and slow-moving? The giant isopod, with its cultural cachet and its inherent strangeness, might be one of the answers. If people can develop affection for a deep-sea crustacean, based on aquarium visits and plush toys, perhaps that affection can be channeled into broader awareness of deep-sea conservation issues. But that is, admittedly, a hope, rather than a certainty. Let me return to the science for a moment, because there is one more aspect of the giant isopod's biology that I find particularly compelling and that I have not yet discussed in detail. It concerns the animal's relationship with the broader isopod order and what that relationship tells us about the versatility of the isopod body plan. Isopods are one of the most diverse groups of crustaceans on Earth. The order Isopoda contains over 10,000 described species, found in virtually every habitat on the planet. They live in the deep ocean. They live in freshwater streams. They live in caves, in deserts, in rainforests, in the soil of your garden. They are found from the equator to the poles, from sea level to mountain tops, from the haddle zone of the ocean to the leaf litter of temperate forests. This is an extraordinary range. Very few animal orders span the full breadth of terrestrial and marine environments the way isopods do, and within this range, isopods have evolved an astonishing diversity of lifestyles. There are parasitic isopods, like the tongue-eating louse, Cimothoa exigua, which enters a fish's mouth, destroys its tongue, and then attaches itself in the tongue's place, feeding on the fish's blood and mucus for the rest of its life. There are wood-boring isopods, like Limnoria, which tunnel into submerged timber and were historically a serious threat to wooden ship hulls and wharf pilings. There are cave-dwelling isopods that have lost their eyes entirely, having evolved in total darkness for so long that vision became unnecessary. There are desert isopods that have evolved the ability to conserve water with an efficiency that rivals insects. And then there are the giant isopods, the deep-sea specialists, the ones that took the basic isopod body plan and scaled it up to massive proportions, filling it with fat, turbocharging its digestive system, and equipping it with some of the most sophisticated eyes in the deep ocean. The fact that the same body plan can produce a 1 centimetre cave-dwelling eyeless scavenger, a 3 centimetre parasitic tongue replacement, and a 76 centimetre deep-sea giant with 4,000 facet compound eyes is a testament to the extraordinary evolutionary flexibility of the Isopod design. It is a chassis that can be modified for almost any environment and almost any lifestyle. And the giant Isopod is not just the largest modification, it is arguably the most extreme. There is a concept in evolutionary biology called the body plan constraint. The idea is that every animal group has a basic body plan, a fundamental architectural template that limits how far evolution can modify the organism. Mammals have a basic body plan, 4 limbs, a spine, internal skeleton, live birth in most species. This plan constrains what mammals can become. A mammal cannot evolve 6 legs. A mammal cannot evolve an exoskeleton. The template sets the boundaries. Isopods have their own body plan, segmented body, exoskeleton, multiple pairs of similar legs, compound eyes, bremous appendages. But within those constraints, the diversity is remarkable. The Isopod body plan is, for lack of a better word, permissive. It allows an unusual degree of modification, and the giant Isopod represents the furthest that modification has been pushed in the direction of size, endurance, and deep-sea specialization. I find it worth noting that isopods are also one of the few crustacean groups that have successfully colonized land. Most crustaceans are strictly aquatic. Crabs, lobsters, shrimp, barnacles, copepods. They live in the water and die if removed from it for extended periods, but isopods made the jump. Woodlice breathe through modified pleopods that retain moisture from the air, functioning as primitive lungs. They're not perfectly adapted to terrestrial life, which is why you find them in damp, dark places rather than in open desert. But they made it. They crossed the boundary that almost no other crustacean group has managed to cross. This success on land, combined with the giant isopod's success in the deep ocean, makes the isopod order one of the most versatile animal groups on Earth. They have conquered the deepest ocean trenches in the driest islands. They have invaded the bodies of other animals as parasites. They have tunneled into the wood of human structures. They have survived mass extinctions and climatic upheavals, spanning hundreds of millions of years. And through all of it, the basic body plan has remained recognizable, segmented, armored, multi-legged, patient. If I were asked to choose a single animal group as the most underappreciated success story in the history of life on Earth, I would choose isopods. Not because any individual species is the most impressive, though the giant isopod comes close, but because the order as a whole has achieved a breadth of ecological success that very few animal groups can match, and it has done so quietly, in the shadows, under rocks and at the bottom of the ocean, without the attention or admiration that we lavish on mammals, birds and the other charismatic groups. The giant isopod is the ambassador of this quiet success. It is the member of the family that gets noticed, the one with the dramatic size, the eerie eyes, the aquarium fame. But behind it stands an entire order of organisms that has been quietly succeeding on this planet for over 300 million years, in more environments and in more ways than almost any other animal group alive. Let me take you even deeper now, into a topic that the 2022 genome study opened up but that has not received as much attention as it deserves. The transposable elements, the jumping genes, because I think these are the key to understanding not just how the giant isopod got so big, but how it has managed to survive for so long. As we discussed, 84% of the giant isopod's genome consists of transposable elements. To put that number in context, roughly 45% of the human genome is composed of transposable elements or their remnants, so the isopod has nearly twice the proportion. But the significance is not just in the quantity, it is in what these elements do. Transposable elements are, at their simplest, sequences of DNA that can move within the genome. Some types copy themselves and paste the copy into a new location, increasing the total amount of DNA. Others cut themselves out of one location and insert themselves into another. Either way, when a transposable element moves, It can disrupt existing genes, create new gene combinations, or alter the regulation of nearby genes. In other words, transposable elements are a source of genetic variation. They introduce novelty into the genome. In many organisms, transposable elements are viewed as genomic parasites. They replicate themselves at the expense of the host genome, and the mutations they cause are more often harmful than beneficial. But in some lineages, transposable elements have been co-opted by the host genome for beneficial purposes. They have been domesticated, in a sense, turned from parasites into tools. The 2022 study suggested that this is what has happened in the giant isopod. The massive proliferation of transposable elements has given the genome an unusual degree of plasticity, the ability to generate new genetic variations rapidly. In a stable environment, this plasticity might not be necessary. But the deep ocean, despite being stable in the short term, has experienced significant changes over geological time. Ice ages have altered the temperature and chemistry of deep waters. Tectonic events have opened and closed ocean basins. The productivity of surface waters, which determines the amount of marine snow reaching the bottom, has fluctuated dramatically over millions of years. An organism with a rigid conservative genome might struggle to adapt to these slow but significant changes. An organism with a highly plastic genome, one rich in transposable elements and therefore capable of generating new genetic variations at a higher rate, would be better positioned to evolve in response. The giant isopod's genome, with its 84% transposable element content, is essentially a genome optimized for long-term adaptability. It is designed to change, even if the changes are slow and invisible to the eye. This is a beautiful paradox. The giant isopod looks like it has not changed in 160 million years. Its external appearance is virtually identical to its ancient ancestors, but beneath that unchanging exterior, its genome has been churning with activity, generating new variations, testing new gene combinations, refining the internal machinery that makes the body plan work. The isopod's stability is not the result of stagnation. It is the result of continuous invisible adaptation. The body stays the same because the genome is constantly adjusting the machinery inside it to keep it working perfectly in a slowly changing world. I find this to be one of the most profound lessons from the giant isopod genome study. Stability and change are not opposites. They can coexist. An organism can look the same for 160 million years and still be evolving, still be adapting, still be changing at the molecular level. The exterior is the visible product of evolution. The genome is the invisible engine, and in the giant isopod that engine has been running, quietly and effectively, for longer than most animal lineages have existed. There is a question that has been floating beneath the surface of everything we have discussed so far, and I think it is time to address it directly. If we were so wrong about the giant isopod, what else might we be wrong about? This is not a rhetorical question. It is not meant to be provocative or alarmist. It is a genuine scientific concern, and it is one that deep-sea biologists think about constantly. The deep ocean is full of organisms that we have classified and described based on the same kinds of limited evidence that led us astray with the giant isopod. Dead specimens, fragmentary observations, inferences drawn from anatomy. And for many of these organisms we may be carrying the same kinds of fundamental errors that persisted in isopod biology for over a century. Consider the giant squid. For centuries, the giant squid existed primarily as a creature of myth and sailors' tales. It was the kraken. It was the monster that wrapped its tentacles around ships and dragged them beneath the waves. When science finally caught up with the legend, we learned that the giant squid was a real animal, a deep-water cephalopod that could grow to enormous size. But for decades, virtually everything we knew about the giant squid came from dead specimens, washed ashore, or pulled from the stomachs of sperm whales. We described its anatomy, we measured its tentacles, we examined its beak, and we made assumptions about its behaviour based on this evidence. It was not until 2004 that the first photograph of a living giant squid in its natural habitat was captured by Japanese researchers using a baited camera system, and it was not until 2012 that the first video of a living giant squid in the deep ocean was recorded. When we finally saw the animal alive, it was different from what we had expected. It was more active, more agile, more deliberate in its movements. The dead specimens on the beach, with their limp tentacles and damaged bodies, had given us a misleading picture of a dynamic, powerful predator. The parallels with the giant isopod are obvious. In both cases, our understanding was shaped by dead specimens and limited observations. In both cases, the living animal turned out to be more complex, more active, and more interesting than the dead specimens suggested. And in both cases, it took modern technology, underwater cameras, genetic sequencing, long-duration observation platforms to reveal the truth. Now consider how many deep-sea organisms we have not yet observed alive. The answer is most of them. The deep sea contains thousands of described species and an unknown number of undescribed ones. For the vast majority, our understanding is based on the same kind of evidence that misled us about the giant isopod. Dead specimens, trawl catches, preserved museum collections. We have no video of their behaviour. We have no genetic data. We have no idea what they actually do in their natural environment. This is not a criticism of the scientists who have done this work. They did the best they could with the tools available to them, but it is a reminder that our picture of deep-sea life is, at best, a rough sketch. The outlines are there, some of the details are filled in, but vast areas remain blank, and some of the details we have filled in may turn out to be wrong. The giant isopod, in this sense, is a gift to science, not because of what it is, but because of what it teaches us about how science works. It is a case study in the dangers of complacent consensus. For over 100 years the consensus was clear. The giant isopod is a scavenger. End of story. And the consensus was wrong, not because the scientists were careless, but because the consensus was built on incomplete evidence, and nobody had a strong enough reason to question it until new tools made the truth visible. This is how science works at its best and at its worst. At its best, science is self-correcting. New evidence overturns old assumptions. New tools reveal new truths. The genome study, the species discovery, the behavioural footage. These are all examples of science correcting itself. The system worked, eventually. At its worst, science is conservative. Once a consensus forms, it acquires a kind of inertia. New evidence that contradicts the consensus is met with skepticism. Researchers who challenge established views face institutional resistance, and for organisms like the giant isopod, which live in environments that are expensive and difficult to study, the consensus can persist for decades or even centuries, simply because nobody has the resources or the motivation to challenge it. I bring this up not to be cynical about science, which I'm not, but to emphasize the importance of the work that remains to be done. The deep ocean is still largely unexplored. The organisms that live there are still largely misunderstood. And every time we send a submersible to the bottom, every time we deploy a baited camera, every time we sequence the genome of a deep-sea organism, we are adding a piece to a puzzle that is still overwhelmingly incomplete. The giant isopod should make us humble about what we claim to know, and it should make us eager to learn more. Let me dwell a bit longer on the specific environment where the giant isopod lives, because I think the more clearly we can picture it, the more we can appreciate what this animal has achieved. Imagine you are standing on the edge of a continental shelf. Beneath your feet the seafloor slopes gently away from the coast, covered in a layer of sediment, sand, mud, the accumulated debris of millions of years of erosion and biological activity. This shallow shelf extends for some distance, perhaps tens or hundreds of kilometres, at depths of a few 100 metres or less. Sunlight still reaches here, at least in attenuated form. There is plant life, there are fish, there are coral formations. This is the ocean that most people think of when they think of the sea. But at the edge of the continental shelf, The floor drops away. The slope steepens dramatically. The sediment becomes finer. The water temperature falls. The light fades. You are entering the Barfield zone, the region between approximately 200 meters and 2,000 meters deep. This is where the giant isopod lives. The Barfield zone is a transitional realm. It is no longer the sunlit shallows, but it is not yet the abyssal plain, the vast flat featureless desert that stretches across the deep ocean floor at depths of 3,000 to 6,000 meters. The Barfield zone has topography. Underwater canyons, seamounts, ridges, slopes. The seafloor here is varied and textured. Currents move along the slopes, carrying nutrients and oxygen. The sediment is soft enough for burrowing, and the temperature, while cold, is within the range that many organisms find tolerable. For the giant isopod, the Barfield zone offers several advantages. Food is more available here than in the deeper abyss, because the continental slopes intercept organic material sinking from the productive surface waters above. Dead fish, dead plankton, and other organic debris accumulate on the slopes and in the submarine canyons, creating patches of relative abundance in an otherwise food-poor environment. The slopes also provide a diversity of microhabitats, soft sediment for the brooding females to bury themselves in, rocky outcrops for shelter, and varying degrees of current that distribute food and chemical signals across the landscape. The giant isopod has been found most commonly at depths between approximately 370 meters and more than 2,000 meters, though the exact range varies by species. Bifidobus giganteus, the largest and most well-known species, is most commonly found in the Gulf of Mexico, at depth between about 370 meters and 700 meters, though it has been recorded much deeper. Other species occupy different depth ranges in different ocean basins. Within its range, the giant isopod encounters a community of organisms that is both sparse and diverse. Brittle stars cover vast areas of the deep-sea floor, their long, flexible arms sweeping the sediment for food particles. Deep-sea anemones anchor themselves to rocks and filter small organisms from the current. Rat-tailed fish, also known as grenadiers, are among the most common fish in the Barfield zone, and they are likely both competitors and occasional prey for giant isopods. Sea cucumbers process the sediment in enormous quantities, digesting the organic material and excreting the cleaned mud. Sponges, some of them glass sponges with skeletons made of silicon dioxide, filter bacteria and dissolved organic matter from the water. It is a community operating at low speed and low energy. The metabolic rates of deep-sea organisms are, on average, significantly lower than those of their shallow water counterparts. Everything is slower. Growth is slower. Reproduction is slower. Movement is slower. Death is slower. The deep ocean runs on a clock that barely moves compared to the frenetic pace of life in a tropical reef or a temperate forest. The giant isopod fits perfectly into this slow world. Its metabolic rate is low enough to survive years without food. Its growth is slow but continuous, adding mass over a lifetime that may span decades, though we do not know the exact lifespan of any giant isopod species with certainty. Its reproduction is infrequent and energy-intensive, producing a small number of large, well-developed offspring rather than a flood of tiny, vulnerable larvae. Everything about the animal is calibrated for a world where energy is scarce, time is abundant, and patience is the ultimate survival strategy. I have spent some time now talking about the threats that the giant isopod faces: plastic pollution, deep-sea trawling, ocean warming. But I want to be careful not to leave the impression that the giant isopod is fragile. It is not. This is an animal that has survived for 160 million years, through conditions that killed the vast majority of species that have ever lived. It has a genome specifically designed for adaptability. It has a body plan that has proven itself across geological time. And it lives in an environment that, while changing, is changing more slowly than the surface world. The giant isopod is resilient. It is among the most resilient organisms on the planet. But resilience has limits. The current rate of environmental change, driven primarily by human activity, is faster than anything the deep ocean has experienced in millions of years. The question is not whether the giant isopod can adapt. The question is whether it can adapt fast enough. This is the central tension in deep-sea conservation more broadly. The organisms of the deep are ancient, specialized, and slow to change. The pressures they face are modern, accelerating and driven by activities that occur far above them on the surface. Plastic waste produced in cities thousands of miles away sinks to the deep floor. Carbon dioxide absorbed by the ocean at the surface slowly acidifies the deep waters below. Heat absorbed at the surface gradually warms the depths. The deep ocean is, in ecological terms, downstream of everything we do on land and at the surface. It receives the consequences of our actions, diluted and delayed but inescapable. The giant isopod cannot lobby for environmental protections. It cannot migrate to cleaner waters. It cannot change its diet to avoid plastic. It can only do what it has always done: sit in the dark, conserve energy, and wait. And whether the world we are creating is one that still has a place for it is a question that only we can answer. But I do not want to end on that note. Not because the threats are not real, but because the giant isopod story is ultimately not a story about decline. It is a story about discovery, about the humbling realization that an animal we thought we understood, one of the most recognizable creatures in the deep ocean, was far stranger, far more complex, and far more sophisticated than we knew. The genome that is nearly twice the size of a human's, the stomach that fills 2/3 of the body, the eyes with 4,000 facets and a reflective layer borrowed from the design of night-hunting predators, the ability to survive 5 years without eating, the reproductive strategy that makes the mothers invisible, the species that hid in an aquarium for years before anyone noticed it was not what they thought, the footage of an isopod eating a shark's face. Every one of these revelations challenged something we assumed to be true, and every one of them made the giant isopod more interesting, not less. The real animal is more remarkable than the textbook version ever was. The real animal is a genetic masterwork, refined over geological time for one of the most demanding environments on Earth, and the fact that it took us over 140 years to begin to see it clearly is not a failure of science. It is a measure of how much we still have to learn. There is a thought experiment I want to walk through with you. It is not a scientific experiment. It is an exercise in perspective, and I think it is worth doing here, near the end of this story, because it captures something about the giant isopod that facts and figures alone cannot convey. Imagine that you are a giant isopod, not in a whimsical cartoon sense, in a literal biological sense. Imagine that your body is armoured and cold, that your blood, if you can call it blood, is a pale fluid called hemolymph, that is copper-based rather than iron-based, which means it is not red but faintly blue when oxygenated. Imagine that you have 14 legs, each one articulated and segmented, each one capable of gripping the soft sediment of the ocean floor. Imagine that your eyes, which sit wide apart on the front of your head, contain nearly 4,000 individual optical units, each one backed by a mirror that doubles the available light. Your visual world is not the bright, colourful world that humans experience. It is a world of faint shapes and shadows, of dim bioluminescent flashes in the distance, of gradients of darkness that convey information, the way colour conveys information to us. Imagine that your primary sense is not vision at all, it is chemistry. Your 2 pairs of antennae, long and sensitive, are constantly sampling the water around you, detecting dissolved molecules at concentrations so low that no human instrument can match them. You live in a world defined by scent, the scent of a dead fish half a kilometre away, the scent of a living organism nearby, the scent of the sediment, with its complex chemistry of minerals, bacteria, and decomposing organic matter. Imagine that you have not eaten in 2 years. Your fat body, the network of lipid-rich tissues distributed throughout your body cavity, has been slowly depleting, calorie by calorie, supporting the bare minimum of biological function. Your metabolism has slowed to a crawl, your heart beats slowly. Your breathing, the gentle movement of your pleopods extracting oxygen from the cold water, is barely perceptible. You are not hungry in the way a human gets hungry. You are not suffering. You are simply in a state of metabolic suspension, a lipo operating at the lowest possible setting, like a machine in standby mode, and then distantly, impossibly faintly, your antennae pick up something, a chemical trace, organic, animal, dead. Somewhere in the darkness, perhaps hundreds of meters away, something has settled to the bottom. A fish carcass, maybe, or a piece of a whale. The chemical plume is thin and scattered, dispersed by the slow deep-sea currents, but it is there. And your antennae, each one a precision instrument honed by 160 million years of evolution, have detected it. You begin to move, slowly. Your 14 legs engage the sediment, one pair at a time, pulling you forward with the deliberate, unhurried gait of an animal that has nowhere to rush to and no energy to waste on speed. The chemical signal grows stronger as you approach. Other isopods are converging too. You sense them in the water, their own chemical signatures mixing with the signal from the food source. There is no aggression. There is no competition in the sense that surface animals experience competition. There is enough. There is almost never enough, but when there is, the deep sea's most patient inhabitants converge and share. You reach the carcass. It is a large fish, perhaps a deep-sea rat-tail, that died and settled to the bottom. Your 4 sets of jaws engage the flesh. You eat, not delicately, not slowly. You eat with the focused intensity of an animal that has not eaten in 2 years and does not know when it will eat again. Your stomach expands, your body swells. The genetically optimized digestive system begins the process of extracting every possible calorie from the meal. Glycolysis pathways fire. Vesicular transport systems shuttle nutrients to storage. The fat body begins to rebuild its reserves, and when the carcass is consumed, when nothing remains but bone and cartilage, you settle back to the seafloor. Your antennae extend, your eyes scan the darkness, and you wait, again, for as long as it takes. This is what it means to be a giant isopod, not a passive existence, not a boring existence, an existence defined by patience, efficiency, and an almost incomprehensible relationship with time. An existence in which the fundamental unit of experience is not the hour or the day, but the season, the year, the decade. An existence in which the most important skill is not strength or speed or intelligence, but the ability to endure. I think there is something profoundly calming about this. In a world that rewards urgency, that celebrates speed and novelty and constant stimulation, the giant isopod offers a counter-narrative. It says that there is another way to be alive, a way that values stillness over motion, patience over urgency, efficiency over excess. The isopod does not strive. It does not compete in the way that surface organisms compete. It simply persists, and it has been persisting successfully for longer than there have been flowers or birds or mammals on this planet. Let me come back to the science one more time, because there are a few loose threads I want to tie up before we close. The question of how many giant isopod species exist is still open. 20 species have been described, but as we discussed, the genus has a documented history of misidentification, and many regions of the deep ocean have not been surveyed with the molecular tools necessary to distinguish cryptic species. The researchers who described Bathynomus yucatanensis explicitly stated that additional undiscovered species likely exist in the Atlantic. The Indo-Pacific, where the majority of species diversity is concentrated, is also incompletely surveyed. The fossil record of the genus is sparse but informative. The oldest known Bathynomus fossils date to approximately 160 million years ago, placing the genus firmly in the Jurassic period. But the broader isopod order is much older, dating back over 300 million years. This means that the lineage that would eventually produce the giant isopod was already established during the Carboniferous period, when the earliest forests were growing on land and the first amphibians were colonizing terrestrial habitats. The fossil isopods from the Jurassic are remarkably similar to modern specimens. The body plan, the segmented armour, the multiple pairs of legs, the broad pleotelson is essentially unchanged. This extraordinary morphological conservatism is consistent with what we know about the stability of the deep-sea environment. When your world does not change much, neither do you, but the genome, as we have seen, tells a different story. The DNA has been churning with activity for hundreds of millions of years, even as the exterior has remained static. The isopod is a living demonstration that evolutionary stasis and evolutionary change can coexist. There is one more aspect of the 2022 genome study that I want to highlight, because I think it deserves more attention. The study found that the giant isopod's genome contains expanded gene families in the hippo-signaling pathway, a genetic pathway that is directly involved in controlling organ size across the animal kingdom. Key genes in this pathway, including DAXUS, protein Kebra, and transcriptional coactivators, were present in significantly more copies in the giant isopod than in other crustaceans. The hippo pathway is one of the most fundamental growth regulation systems in biology. It was first discovered in fruit flies, where mutations in the pathway caused massive overgrowth of organs, prompting researchers to name it after the hippopotamus. The pathway works by regulating cell proliferation and cell death, determining how big an organ grows before the growth signals shut off. In the giant isopod, the expanded gene families in this pathway suggest that the animal's growth regulation system has been specifically modified to produce larger organs and, by extension, a larger body. This is a molecular mechanism for gigantism, not a vague environmental explanation, not a hand-wavy reference to cold water or reduced predation. A specific identifiable genetic modification that directly promotes larger body size. Combined with the thyroid and insulin hormone expansions, the suppressed lipid degradation, and the supercharged digestive system, it paints a picture of an organism whose genome is not just permissive of large body size but actively driving it. This matters beyond the isopod itself, because deep-sea gigantism is observed across many different animal groups: giant squid, giant spider crabs, giant amphipods, giant tube worms. If the giant isopod's gigantism is driven by specific identifiable genetic pathways, then similar pathways may be at work in other deep-sea giants. The isopod genome may turn out to be a Rosetta Stone for understanding gigantism across the deep ocean. It may reveal that what looks like a simple ecological trend, big animals in deep water, is actually a convergent genetic strategy, with different lineages independently evolving similar molecular solutions to the same environmental challenges. This is speculative, but it is testable speculation. As more deep-sea genomes are sequenced, as the cost of genomic research continues to fall and the tools become more accessible, we will be able to compare the molecular pathways underlying gigantism across different deep-sea lineages. If the same pathways keep appearing, expanded in similar ways in different organisms that independently evolved large body size in the deep ocean, that would be a remarkable finding. It would suggest that evolution has a limited number of solutions to the problem of growing large in the deep sea, and that different organisms have converged on the same answers. But for now, we have the giant isopod, one genome, one window into the genetic architecture of deep-sea gigantism, and what that window reveals is enough to overturn a century of assumptions. I want to close with something that is not a fact or a study or a piece of evidence. It is a feeling, and I think it is the right feeling to close on, given the nature of this story. There is an immensity to the deep ocean that is difficult to comprehend, not just its physical size, though that is staggering, but the temporal immensity. The deep ocean has been there, essentially as it is now, for hundreds of millions of years. The organisms that inhabit it have been there for comparable spans of time. The giant isopod has been on the bottom of the ocean, in the dark, in the cold, for 160 million years. It was there before the asteroid that killed the dinosaurs. It was there before the Himalayas existed. It was there before the first primate walked on its hind legs. It was there before the Atlantic Ocean opened. It was there before the Indian subcontinent crashed into Asia. It has been there, doing what it does, with the patience and efficiency of an organism that is perfectly matched to its world, for longer than the human mind can easily grasp. And tonight, right now, at this very moment, somewhere in the Gulf of Mexico or in the waters off Australia, or in the deep Pacific or Indian Ocean, giant isopods are doing what they have always done. Some of them are sitting on the seafloor, their antennae extended, sampling the chemistry of the water. Some of them are walking slowly toward a distant chemical signal, converging on a food source that fell from the surface world hours or days ago. Some of them are feeding, their 4 sets of jaws working through flesh and bone and cartilage, their stomachs expanding to fill their armoured bodies. And somewhere, buried in the soft sediment, invisible to every instrument we possess, a female is brooding her eggs. She has not eaten in months. She will not eat for months more. She is in the dark, in the cold, under immense pressure, protecting the next generation of an animal that has been protecting its next generation in the same way since the Jurassic. She does not know that we exist. She does not know about our cities, our technology, our science, our plastic. She does not know that somewhere far above her, on the surface of an ocean she will never see, a species that has existed for a fraction of her lineage's history is altering the chemistry of the water around her, warming the temperature of her world, and filling her food supply with indigestible synthetic debris. She simply waits, and she will keep waiting, because that is what she was built to do, and what her ancestors have done for 160 million years. The giant isopod does not need us. It does not benefit from our attention. It has survived without us for longer than we have existed as a species, but we might benefit from paying attention to it, from sitting with the humility of realizing that an animal we thought we understood was far more complex than we knew, from recognizing that the largest habitat on our planet is still largely a mystery, from understanding that patience, efficiency, and quiet persistence are strategies that have proven themselves across geological time, even if they are not the strategies we celebrate. We were wrong about the giant isopod, and that is okay. Being wrong is where science starts. Being willing to be wrong is where wisdom starts, and the deep ocean, with its ancient, patient, armoured inhabitants, has plenty of both left to teach us. Sleep well tonight. And if you find yourself wondering what is happening in the darkness at the bottom of the ocean, know this: the isopods are there. They have always been there, and they are doing just fine. Actually, before we drift off, let me tell you one more thing, because I think there is a story within the story that we have not fully explored, and it has to do with the way we discover new species in the deep sea, and what those discoveries tell us about the limits of human observation. When Huang Ming-Chi sequenced the DNA of that isopod at the Enoshima Aquarium, the one that turned out to be Bathinomus yucatanensis, rather than Bathinomus giganteus, he was not looking for a new species. He was conducting a routine comparative study. He expected the sequences to match. He expected confirmation of what everyone already believed. The discovery of a new species was, in the strictest sense, an accident. This is remarkably common in taxonomy, particularly in deep-sea biology. The history of species discovery in the ocean is littered with accidental findings, with organisms that were sitting in museum jars, misidentified in aquarium tanks, or mislabeled in research collections for years or decades before someone happened to look at them with the right tools or the right question. The tools in this case were molecular, genetic sequencing, a technology that did not exist when most deep-sea specimens were originally collected. Think about what this implies. We have museum collections of deep-sea organisms that stretch back over a century. thousands upon thousands of specimens collected from expeditions around the world, preserved in formalin or ethanol, labelled and catalogued according to the best taxonomy of their time. And within those collections there are almost certainly additional undescribed species, organisms that were collected, examined, classified as a known species based on external appearance, and filed away, waiting for the technology that could reveal them to be developed. The molecular revolution in taxonomy has, over the past few decades, been steadily uncovering these hidden species. In group after group, from fish to crustaceans to worms to mollusks, genetic analysis has revealed that what was thought to be a single, widespread species is actually a complex of closely related but distinct species, each occupying its own geographic range or depth zone. The technical term for these are cryptic species, species that are morphologically indistinguishable or nearly so, but genetically distinct. Giant isopods are a textbook example of the cryptic species problem. Baphinomus yucatanensis looks almost exactly like Baphinomus giganteus. The differences are subtle, slightly more slender body proportions, slightly longer antennae, a slightly different shell colour, a different shape to the thorax. These are not the kinds of differences that jump out at you when you are examining a preserved specimen under a microscope. They are the kinds of differences you notice only when you are specifically looking for them, armed with the knowledge that they might exist. And for over a century, nobody was looking. The cryptic species problem extends far beyond isopods. It is one of the central challenges in modern marine taxonomy, and it has profound implications for conservation. If we do not know how many species exist, we cannot accurately assess which ones are threatened. If we are lumping multiple species into a single classification, we may underestimate the true diversity of an ecosystem and overestimate the resilience of individual species. A species that appears to be widespread and abundant may actually be several species, each of which is narrowly distributed and vulnerable. Without molecular data, we are flying blind. For the giant isopod, the practical implications are significant. The researchers who describe Baphinomus yucatanensis pointed out that giant isopods are targets of deep-sea trawl fisheries in some regions. If fisheries managers believe they are harvesting a single, widespread, abundant species, they may set catch limits that are too high for individual species within the complex. A fishing operation that harvests Baphinomus giganteus and Baphinomus yucatanensis indiscriminately, without distinguishing between them, may be overexploiting one or both species without realizing it. And since giant isopods reproduce slowly, with small clutches and long brooding periods, even modest overharvesting can have outsized effects. This is not a hypothetical concern. Deep-sea trawling operations have expanded significantly in recent decades, driven by the depletion of shallow water fish stocks and the development of technologies that allow commercial fishing at greater depths. The bathyal zone, where giant isopods live, is increasingly within the reach of commercial fisheries, and the regulatory frameworks for deep-sea fishing are, in many regions, either non-existent or inadequate. I am not suggesting that giant isopods are on the brink of extinction. We do not have the data to make that claim, and it is important to be honest about what we do not know. But I am suggesting that the combination of slow reproduction, cryptic species diversity, expanding fisheries pressure, plastic contamination, and ocean warming creates a set of risks that deserves attention. And the first step toward addressing those risks is acknowledging how much we still do not know. Let me bring this back to where we started. The title of this piece is "We Were Wrong About the Giant Isopod," and as we have seen, we were wrong in multiple ways. We were wrong about its biology. Its genome revealed an organism of extraordinary genetic complexity, not a simple scavenger, but a precisely engineered survival machine. We were wrong about its behaviour. The footage of an isopod eating a shark's face shattered the passive scavenger narrative. We were wrong about its taxonomy. A new species was hiding in plain sight for over a century, in a museum tank, in a well-studied group of animals. unrecognised until molecular tools revealed the truth, and we were wrong about its simplicity. The giant isopod is not simple. It is not primitive. It is an organism that has been refining its survival strategy for 160 million years, using a genome nearly twice the size of ours, carrying genetic machinery for growth regulation, fat storage and digestive efficiency that would impress any engineer. But here is the thing about being wrong. Being wrong is not the end of the story. It is the beginning. Every correction opens a new chapter. Every disproven assumption reveals a deeper truth. The giant isopod that emerges from the corrected science is more fascinating, more complex, and more worthy of study than the oversimplified version that preceded it. We did not lose anything by being wrong. We gained a richer, more accurate, and more wondrous picture of a remarkable animal, and the corrections are not finished. There are almost certainly more giant isopod species waiting to be discovered. There are almost certainly behavioural patterns that we have not yet observed. The reproductive biology remains a near-total mystery. The interactions between species, the population dynamics, the lifespan, the growth rate, the sensory world, the social behaviour, if any, all of these remain largely unknown. The 2022 genome study was a beginning, not an end. It opened doors. It did not close them. The deep ocean keeps its secret slowly. It does not yield to impatience. It requires the same kind of patient, persistent long-term commitment that the isopod itself embodies. We must send submersibles to the bottom. We must deploy cameras and leave them for months. We must collect specimens carefully and analyse them with the best available tools. We must resist the temptation to assume we already know the answer. And we must be willing, always willing, to discover that we were wrong. The giant isopod has been teaching this lesson for 160 million years. It has been sitting on the bottom of the ocean, in the dark under crushing pressure, with a genome full of surprises and a lifestyle that defies our expectations, waiting for us to pay attention. And now that we are finally beginning to look, really look, with the right tools and the right questions, what we are finding is extraordinary. Tonight the isopods are in the dark. They are on the bottom. They are doing what they have always done, and they will be there tomorrow, and the next day and the next, long after we have turned our attention to something else. That is what they do. That is what they have always done. And that, in the end, is what makes them remarkable. Not their size, not their armour, not even their genome, but their patience, their persistence, their quiet, relentless, unshakable commitment to being exactly what they are, in exactly the place they belong for as long as the ocean will have them. Good night, and may your rest be as deep and as still as the ocean floor. There is one final thought I want to leave you with. It concerns the relationship between knowledge and wonder, and I think the giant isopod illustrates it perfectly. When we knew less about the giant isopod, when it was just a large deep-sea scavenger that sat on the bottom and ate dead things, it was interesting. It was a curiosity, something to show in a museum display and talk about at dinner parties. A fun fact. Did you know there's a giant roly-poly at the bottom of the ocean? Interesting, yes, but not awe-inspiring. Not the kind of thing that keeps you thinking for days. Now consider what we know today. An organism with a genome nearly twice the size of a human's, 84% of it, composed of jumping genes that give it extraordinary evolutionary flexibility. A creature whose growth is driven by expanded thyroid and insulin hormone pathways, whose fat metabolism is actively suppressed to preserve energy reserves, whose stomach fills 2/3 of its body and is backed by genetically supercharged digestive machinery. An animal that can fast for 5 years, that gives birth to fully formed young in a pouch on its underside, that has 4,000 facet compound eyes with a reflective mirror layer, that has been filmed eating the face of a living shark, that contains at least one species that hid in an aquarium for years without being recognized, that has survived every mass extinction event for 160 million years. This is more interesting, not less. The more we learn, the more extraordinary, the animal becomes, and this is the point I want to make. Knowledge does not diminish wonder, it amplifies it. The giant isopod is not less mysterious because we sequenced its genome. It is more mysterious because the genome revealed layers of complexity we had not imagined. Every question we answer opens 3 more questions. Every discovery reveals new depths of ignorance. This is the beautiful paradox of science. The more you know, the more you realize how much you do not know. The deep ocean is full of organisms like this, animals and microbes and plants that seem simple from a distance, but reveal staggering complexity up close. The 6-gill shark, which has been cruising the deep for over 200 million years, longer even than the giant isopod. The vampire squid, which is not a squid and not an octopus, but something in between. A living relic of a lineage that diverged from both over 160 million years ago. The barrel fish, whose transparent head allows its tubular eyes to rotate upward and forward, scanning the water above for the silhouettes of prey against the faint glow of the surface. The zombie worm, Osidax, which has no mouth and no stomach, but dissolves whale bones with acid secreted from its skin and uses symbiotic bacteria to extract nutrients from the dissolved bone. Each of these organisms carries its own set of surprises. Each of them has been studied to some extent, and each of them has revealed unexpected complexity, and each of them lives in the same vast, dark, cold habitat as the giant isopod, in the deep ocean that we have barely begun to explore. We are at the beginning of a golden age of deep-sea discovery. The tools available to marine biologists today: autonomous underwater vehicles, long-duration benthic observatories, high-throughput genomic sequencing, environmental DNA sampling are more powerful than anything that has existed before. In the coming decades, we will learn more about the deep ocean than we have learned in all of prior human history, and some of what we learn will surprise us. Some of it will correct assumptions we did not even know we were making. Some of it will reveal organisms and processes that are genuinely without precedent. The giant isopod showed us what this looks like. It showed us what happens when new tools meet old assumptions, and the result was not just a correction, it was a revelation. A revelation that the deep ocean is not the barren, lifeless, simple place that early scientists imagined. It is a complex, dynamic, ancient ecosystem full of organisms that have been solving the problems of survival for longer than most lineages on Earth have existed. And the isopod, sitting there in the dark, with its 4,000 eyes and its jumping genes and its 2/3 stomach and its infinite patience, is proof that the most extraordinary things are often the ones we overlook, the ones we think we already understand, the ones we label simple and file away, the ones that, when we finally look closely enough and honestly enough, turn out to be anything but. That is the lesson of the giant isopod, and it is a lesson that applies far beyond biology, in science, in life, in the way we approach the unknown. The things we think we understand are often the things we understand least, and the willingness to say we were wrong is the beginning of every truly important discovery. The deep is patient, the deep has time, and the deep has more to teach us than we have yet learned to ask. Rest now. The ocean is quiet, the isopods are still, and tomorrow the world will still be full of mysteries, waiting, as they have always waited, for someone to look closely enough to see them. And if you are still listening, still awake, still with me here at the end of this long journey through the dark waters of the deep ocean, I want you to know something. The giant isopod does not care about any of this. It does not care about genomes or taxonomy or conservation debates. It does not care about Shark Week footage or Japanese aquariums or plush toys made in its image. It does not care that we were wrong about it for 140 years, and it does not care that we are now, slowly, beginning to get it right. It is an animal, a crustacean, an ancient, armoured, patient, perfectly adapted machine for surviving in one of the harshest environments on Earth. It does not need our understanding. It does not need our admiration. It does not need anything from us at all. But we need something from it. We need the reminder that the world is bigger, stranger, older and more complex than we typically imagine. We need the humility that comes from realizing how much we have gotten wrong about a single well-known animal. We need the perspective that comes from contemplating a creature that has been alive on this planet for 65 times longer than our entire species has existed. And perhaps most of all, we need the calm that comes from knowing that somewhere, right now, in the cold darkness at the bottom of the ocean, something ancient is resting. Something that has survived everything this planet has thrown at it. Something that will, in all likelihood, be there long after our cities have crumbled and our names have been forgotten. The Isopod does not hurry. The Isopod does not worry. The Isopod waits. And in the waiting there is a kind of wisdom that we, with all our intelligence and all our technology and all our restless, urgent, anxious energy, might do well to learn from. Good night. The deep is still, and it will still be there in the morning.